Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about linker and adapter. Linkers and adapters are short chemically synthesized DNA sequences that are useful for the ligation of DNA fragments, particularly those having blunt ends. In recombinant DNA technology, very often DNAs are cut into fragments and the fragments are joined with other fragments. DNA molecules are cut by using the enzyme restriction endonuclease. Restriction endonuclease make two types of cuts. One is straight cut and the other is staggered cut. The straight cut produces blunt ends and the staggered cut produces sticky or cohesive ends. The ligation of DNA fragments having sticky or cohesive end is efficient. It is easy, but the ligation of DNA fragments having blunt ends is not efficient. So, the ligation of blunt-ended DNA fragments can be done by using linker and adapter. First of all, we shall discuss about linker. Linkers are short chemically synthesized double-stranded DNA molecules having blunt ends. Linkers possess recognition site for specific restriction enzyme. For example, this is a linker that is known as EcoR1 linker. EcoR1 linker possesses recognition site for the restriction enzyme EcoR1. The arrow mark indicates the recognition site that is made for the restriction endonuclease known as EcoR1. So now we will see that how by using this EcoR1 linker these two DNA fragments which have blunt ends can be ligated easily. So first of all, we have to ligate these two DNA fragments with the same linker by using the enzyme DNA ligase. So this double-stranded DNA molecule is ligated with the EcoR1 linker by using the enzyme DNA ligase. So this double-stranded DNA fragment has the same linker on either side. Similarly, this is another DNA fragment. This DNA fragment is also double-stranded and the ends are blunt ends. This DNA fragment is also ligated with the EcoR1 linker by using the enzyme DNA ligase. After ligating this DNA fragment and this DNA fragment with linker, next step is to treat these DNA fragments with EcoR1. So EcoR1 will recognize the specific site and it will cleave. So the cleavage by EcoR1 will produce sticky or cohesive ends. Sticky or cohesive ends. This is a cohesive end and this is another cohesive end. These two ends are complementary to each other. When these two DNA fragments are mixed, annealing occurs very easily between these two cohesive ends because this cohesive end is complementary to that cohesive end. So base pairing occurs very easily between these two sequences. Because of this, annealing easily takes place. So here there is base pairing between adenine, thymine, adenine, thymine, thymine, adenine and thymine, adenine. After annealing, these two DNA fragments can be easily ligated very efficiently by using the enzyme DNA ligase. But when we are using linker for the ligation of DNA fragment, we should be sure that the DNA fragments do not contain internal restriction site for the same endonuclease, because if we use such type of DNA fragments for ligation by using linker, then because the fragments contain internal recognition site, so the restriction enzyme will also cut this DNA fragment into pieces. Now we shall discuss about adapters. Like linkers, adapters are also short chemically synthesized oligonucleotides that are used for the ligation of DNA fragments having blunt ends. So what is adapter? Adapters are short oligonucleotides that can be single-stranded or it can be double-stranded. And the double-stranded adapter can have two blunt ends. Or the double-stranded adapter can have a blunt end on one side and a sticky or cohesive end on other side. So by using adapters, DNA fragments having blunt ends can be easily ligated. So how this will occur? Let us see. For example, these are the two DNA fragments. This fragment is double-stranded and this fragment is also double-stranded. These two fragments are blunt-ended. So we want to ligate these two DNA fragments by using adapter. So first of all, we have to ligate these two DNA fragments with a specific adapter which is having a sticky or cohesive end on one side. Similarly, ligate this DNA fragment also by using the enzyme DNA ligase with the same adapter. So these single-stranded protruding ends are complementary. So base pairing will occur. Due to base pairing, annealing can occur easily between these two DNA fragments. So after annealing between these two DNA fragments, due to the complementarity of the protruding ends, annealing occur easily. After annealing, ligation can be accomplished by using the enzyme DNA ligase. So this is the process by which we can use adapters for ligation of DNA fragments. But while we are using adapters, there are certain limitations. First limitation is adapters can anneal among themselves to form dimer. This is a dimer that is composed of two adapters. Due to complementarity, these two adapters can join with each other in the presence of enzyme DNA ligase to form a dimer. So when this occurs, ligation of the adapter with the DNA fragment may not be possible. Similarly, adapters can also ligate to themselves. For example, this is the DNA fragment. And this DNA fragment has been ligated with adapters by using the enzyme DNA ligase. So here you see on both sides of these DNA fragments there are several adapters. There are several adapters that have been ligated with the help of enzyme DNA ligase. So to overcome this problem, these adapters are first treated with the enzyme alkaline phosphatase. So alkaline phosphatase is an enzyme that removes the phosphate group that is present at the 5' end. You see, this is the 5' end and this is the 3' end. Similarly, this is the 5' end and this is the 3' end. So when phosphate group is present at the 5' end, phosphodiester bond formation can occur. But when we remove the phosphate group that is present at the 5' end by using the enzyme alkaline phosphatase, formation of phosphodiester bond is not possible. So self-ligation between adapters would not occur. So by using the enzyme alkaline phosphatase, we can prevent the self-ligation of adapters. If the self-ligation of adapters occur in the presence of enzyme ligase, ultimately the DNA fragment will remain blunt-ended. So it will not serve any purpose. So after removing the 5' phosphate group, self-ligation is not possible. When ligation is necessary, for that a phosphate group can be added to the 5' end with the help of enzyme polynucleotide kinase. So friends, this is about linker and adapters, the uses and the limitations that are associated with them. Friends, please share, like and subscribe, and also give your comments. See you, bye bye.